All right, welcome back, fellow scientists. So today is the last video for the flipped classroom. So we're going to end with a bang. We're going to talk about two things that are like absolutely incredible and amazing and yeah, really, really cool. So first thing we're going to talk about is cloning. And before we talk about the process of cloning, let's go ahead and let's just define what a clone is. So a clone is two organisms with the same DNA. So I guess cloning would be making two organisms with the same DNA. So here's a question for you. Do clones occur naturally? Yeah, they do. Any organism that reproduces asexually, such as bacteria or some lizards, they produce clones of themselves. Are there any sexually reproducing organisms that produce clones? Yeah, humans produce clones. Identical twins are technically clones. They're two organisms with the same DNA. All right, so now that we've established that clones happen naturally, now let's go ahead and look at how humans can make clones. So we're going to use sheep as our example because the most famous mammal that has ever been cloned is Dolly the sheep, which happened in 1996, I do believe. And so I'm using this little cheat diagram right here. You can find it online. Just Google cloning or something like that and print it out. Or if you want to try to reproduce this, I'll give you like a 10-second drawing class on how to draw a sheep. So how to draw a sheep, first you draw a cloud, okay? And then you give the cloud legs. And then you draw like a little tiny cloud for a tail. And then you draw like an oval right there. And then you draw another smaller oval for an ear. And then you give it an eye. And then you make it happy like that. So that's a sheep. And now you can see why I am using the diagram instead of my amazing artistic skills. So here's the process of cloning. You start with sheep number one. Okay, so sheep number one could be male or could be female. Doesn't really matter. You take a somatic cell, any kind of cell except for a gamete, out of that sheep, right? You harvest that cell, and this cell is diploid. Right, remember back from genetics, diploid means two sets, one from mom and one from dad. So this has the all the DNA that we need to make a sheep. Okay, so then you take sheep number two and you take an egg cell from sheep number two. So because we're taking an egg cell, obviously sheep number two has to be female, right? So you take an egg cell, and if you'll remember, eggs, gametes, right? Eggs and sperm are gametes, and they're haploid. So they only have one set. So it takes two gametes to make a whole intact entire sheep in this case because we need two sets of chromosomes, two sets of genes, right? So this egg cell is haploid, okay? So then what we do is we take this haploid nucleus and that goes away, and that actually gets thrown away. This is my little version of a garbage can, right? That actually gets thrown away into the garbage, okay? Then you take this diploid cell, and now we have an empty egg and small little electric shock, and now all of a sudden we have an egg. So this is egg with a diploid nucleus. So 2N, it's a diploid egg. Now the normal way we get a diploid egg is through the process of fertilization. But here we used electric shock and we put a diploid nucleus into an empty egg. And so now the egg realizes, oh, I have a diploid nucleus. Okay, well, let's start dividing. I have everything that I need to make a full, entire, intact sheep. So divide, 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 and then at a certain stage, this little sheep embryo gets implanted into sheep number 3, into the uterus. So obviously, sheep number 3 has to be female. And then after the appropriate gestational period for sheep, then this little sheep number 4, this lamb, is born. All right, so here's a question for you. Which sheep which sheep are the clone? Which sheep are the clones of each other? Yeah, you're right. Number 1, right? And number 4. Number 2 and number 3 were just kind of like used in the process, but they didn't contribute any DNA, right? Remember, we took the nucleus out, and then this sheep, even though she carried and gave birth to the clone, she didn't contribute any DNA to the clone. So all of the DNA in sheep number 4 came from sheep number 1. All right, so that's the basic process of cloning, and obviously it's a little bit more complicated. And no, there are no records of man-made what was I going to say? Man-made human clones. Obviously, identical twins are normal human clones. But one of the interesting things is that Dolly did go on to have a normal life for a sheep. She gave birth to 6 lambs. However, she developed arthritis at the age of about 5 years, which sheep live to a grand old age of about 12 years. So developing arthritis at age 5, that's a little bit early. And she died from sheep pulmonary adenomatosis, adenomatosis, yeah, adenomatosis. In 2003, she was 7 years old, right? And usually only sheep that are like 11 or 12 years old die from this disease. So it's almost as if she was born already old. It's almost as if she aged prematurely. And in AP biology, we talk about telomeres, which are little stretches of DNA at the end of chromosomes that get shortened with every round of replication. And so this experiment and others lead some scientists to believe that telomeres are actually very integral in the process of aging. And that even though, right, like Dolly, this cloned lamb right here, was born, her cells, because they came from an adult sheep, her cells were already like 4 or 5 years old at the time she was born. So anyways, that's something kind of cool to think about. So then moving on, so now what can we do? Well, you remember talking about stem cells, and you remember talking about, okay, so if we have a embryo from an in vitro fertilization clinic, we can harvest that inner cell mass and we can expose it to different chemicals, and that's a stem cell, so it's a pluripotent stem cell, so it can turn into many different types of tissue. So we can grow a heart, or we could grow a pancreas, or we could grow, cartilage for an ACL, MCL, something like that. Right? Which is all well and good, but the only problem is that this embryo that we started with doesn't have the same DNA as the person that it's going into. So this person, their immune system is going to attack this foreign tissue, and they have to be on immunosuppressant drugs for the rest of their lives and everything like that. However, there's a process that, to my knowledge, isn't actually in practice, but it's hypothetical, it's theoretical. It's called therapeutic cloning, okay? So therapeutic cloning is basically human cloning, but you don't let the embryo develop more than, 2 weeks, right? So you go through the same process. You have a donor egg, and then you have a person who wants to have a clone of themselves, right? And so then electric shock, and you fuse it, and then you let it develop for 2 weeks. But then, just like with stem cell therapy, then you can take out the inner cell mass, and then you can expose it to different chemicals. And so then we can make either bone or muscle or nervous system or a heart or pancreas or, ACL, MCL, like we did in the last one. And the cool thing about that is now this tissue is the exact same DNA as the donor, as the patient, right? So there are obviously oodles and oodles of ethical dilemmas with that. So stem cells by themselves are ethically charged, but then to bring in human cloning and use it like you would a stem cell, man, I don't know, that's a bridge too far in my opinion. But anyways, different people have different views and ideas, so that's the basic process. So now you're more informed, so now you can be more involved in the discussion. All right, so second process that we're going to talk about is gene therapy. So gene therapy is this amazing process that has the potential to cure genetic diseases. So very quickly, we're going to use sickle cell anemia as our example. So this is what a normal red blood cell looks like. And a person who has sickle cell anemia, some of their red blood cells, they don't make the protein hemoglobin correctly. And so some of their red blood cells are shaped like a sickle. Now, round red blood cells flow very nicely through capillaries and veins and arteries and stuff like that. But sickle-shaped cells don't. They get caught, they get clogged, and they make clots. And that's not good, okay? So we're going to do a little drawing here. So we're going to draw, I know, my artistic skills are amazing. We're going to draw a person. And we're going to say that they have sickle cell anemia. So they're not happy. They're sad. Okay. Now let's draw another person over here, and we're going to say that they do not have sickle cell anemia. So no SCA, no sickle cell anemia, and they are happy. Okay, so what gene therapy looks to do is we can take the DNA from this person that does not have sickle cell anemia. Since we can sequence DNA now, we know exactly where that gene is and what it makes, and we can use restriction enzymes, right? And we can cut out the gene that makes hemoglobin. So we can just write HB, hemoglobin, okay? And then we can take that gene and we can put that gene into a virus. So there's the virus. So here's the gene, and it's going to go into the virus, right? And so now we have this little, like that. So now the gene's in the virus. So now what gene therapy does is gives this virus to the person who has sickle cell anemia. So this virus then is programmed to go and infect the bone marrow, because that's where blood cells are made. Infect the bone marrow of this person with sickle cell anemia. And so then if gene therapy works, now the person has the correct gene so that their cells, their bone marrow, can make hemoglobin correctly. And so now, whoop, they have no sickle cell anemia. And now they're happy, right? So this is actually in clinical trials right now. So let me show you two articles. Let's see, I want this one. And you can look this up. I'm not going to spend time going through all the ins and outs, but scientists are using, researchers, medical doctors are using gene therapy to cure blindness, which is really, really cool. February 8th, 2012, Scientific American. And then they're also, oops, I didn't have this one set up. Here we go. And then they're also using it to fight cancer. So gene therapy shows promise against leukemia and other blood cancers. So this is December 8th, 2013, Health Daily News. The basic idea is leukemia is a disorder of the blood, right? And so scientists take blood cells out, researchers, medical technicians take blood cells out of the patient and then infect them with a virus. So in this scenario, the virus never actually goes into the patient. Blood cells come out, and then virus injects the DNA into the blood cells while the blood cells are outside of the patient. And then those blood cells, just the blood cells, not the virus, just the blood cells go back into the patient, and then they're programmed to either find cancer and destroy cancer, or they're programmed to just, you know, be good blood cells and do what they normally do. So anyways, gene therapy is really, really cool and really, really promising. And yeah, it's awesome. So I hope that you've enjoyed this video. Thanks for watching, and I will see you on the flip side.